All right, hello and welcome to the final section. Look at the tree that's in our textbook and just try to get a bearings again. This represents the whole phylogeny, and you can see there are some questionable areas. Now we have the excavata, which was already discussed, the SAR clade, which includes the chimenophiles, the alveolates, and the rosarians, and then the archaeplastida, which include the red algae and the green algae, which are the closest related to plants. Now we're going to talk about the Unikonta, where we have some close relatives of fungi and animals. So the Unikonta divide up into the Amoebozoans and the Opisthokonts, and then further. So we're going to kind of talk about those. Just kind of back to our idea here. Remember, there are four major supergroups of protists. We've already talked about the excavates, the SAR group, the archaeplastida, and then we're on the Unikonta. So let's do a quick concept map showing the relationships between the groups within the major clade Unikonta. Okay, so we're on the final group, Unikonta. So let's go ahead and draw a concept map type format showing the relationships between the groups within this clade. Really, this isn't even a true clade. Okay. So this is another polyphyletic group. So we have the ancestor, eukaryotic. And then remember, we have the excavates. All right. And then we have the SAR clade. And then the archaeplastida. And finally, we have the group that we'll be working on now, Unikonta. So the Unikonta group is a polyphyletic group, and it's because the fungi and the animals kind of fall into this group, right? So the protists that we'll be dealing with are going to include a couple major groups. Okay, so we have two major groups here. We have the Amoebozoans, and we have the Opisthokonts. Okay, the Amoebozoans split into three major groups. Okay, so we have the slime molds. We have the tubulinids. And then we have the Entamoebas. Now the slime molds further divide into two major groups. We have the Plasmodial slime molds, and then we have the cellular slime molds. All right. So, and then we have what's called the Opisthokonts. Okay, now we're going to do this right here. So these are two major groups of at least protists, right? So we have the nuclearids. And we have the choanoflagellates. Now, these are sister groups to both the fungi and the animals. So nuclearids is most closely related to fungi. Okay, again, a different chapter. And choanoflagellates are basically the direct ancestors of animals. That's going to be a couple chapters at the end of the semester. All right. So we have the Unikonta, which divide into two major groups. We have the Amoebozoans, and we have the Opisthokonts. Okay. And the Amoebozoans split into three groups. We have the slime molds, the tubulinids, and the Entamoebas. And the slime molds further divide into plasmodial slime molds and cellular slime molds. And then the Opisthokonts are kind of divided up into two major categories, including the groups that make it polyphyletic. And so we have the nuclearids, which are the protists, and the choanoflagellates, which are also protists. However, the nuclearids are the direct ancestors of fungi, and the choanoflagellates are the direct ancestors of animals. All right, so let's look at the characteristics of at least the protist groups within the. Okay, so the supergroup Unikonta of protists include animals and fungi. There are two major clades. There are the Amoebozoans and the Opisthokonts, and it's very unclear as to. Let's talk about the Amoebozoans first. They are amoebas, but they're not like the amoebas that we've talked about previously. These amoeba have lobe or tube-shaped pseudopodia, as opposed to the long, thin, thread-like shaped pseudopodia that we've talked about before. The Amoebozoans include the slime molds, the tubulinids, and the entamoebas. The slime molds, or mycetozoans, were thought to be fungi due to the fruiting bodies that are formed during their life cycle. These fruiting bodies produce spores very similar to the structures that are found in fungi. However, they are not fungi, so this resemblance is likely the result of convergent evolution. Slime molds include two lineages, the Plasmodial slime molds and the cellular slime molds, which are quite different from one another. So Plasmodial slime molds are usually brightly colored. You can see some images here. I have one that's orange, and then we've got yellow, but some of them are kind of more red, but this is really like a dark orange. Now, it's important to know that this is a single cell right here. These fruiting bodies, or at least the ones shown here, result from a single cell. All of these are a single-celled organism. It's a very large cell which contains multiple diploid nuclei. So what actually happens is that there's no cytoplasmic division, only nuclear division. Now, the very large mass is called a plasmodium, and this is the feeding stage of the life cycle of a plasmodial slime mold. And it's also important to note that this is not the same Plasmodium falciparum that actually causes malaria. Right? So remember, that's an organism that infects the blood and gets inside the blood cells, and it's transmitted via mosquitoes. This is something completely different. Now, I've provided a video in Canvas. It's I would go ahead and watch it. It's quite interesting to see the process of its life cycle and how. So this is the first part of the life cycle. We can see we're going to start with the feeding plasmodium. So you can see this is a very large mass. It's a single cell with multiple diploid nuclei inside it. It becomes mature, okay, so it moves, and if you watch that video that I provided in Canvas, you'll be able to see how it moves towards a food source. It's pretty smart. Now, once it's happy, it'll eventually start to produce sporangia. Right? So sporangia are structures that are going to emerge from the mature plasmodium and produce spores eventually. So this is more like a mature sporangium. You can see there's a stalk and then a very large spherical structure with cells inside. These cells undergo meiosis to produce spores, which are haploid. So remember, these are diploid, and then after meiosis, now you have haploid cells. The spores are going to be released into the environment, and if the environment is healthy, it will germinate. And it can germinate to produce flagellated cells or amoeboid type cells. Can fuse, which is fertilization, and then develop into a diploid zygote, and then of course mature back to a feeding plasmodium. Cellular slime molds are multicellular organisms. So these actually form multicellular aggregates, which are the aggregates that basically move around like a slug. These cells are individual, so that they feed individually. Okay, so it's almost like a unicellular aggregate or multicellular aggregate of single cells, really. The aggregate forms in order to migrate and find a good feeding source. They do also form fruiting bodies. So let's look at an example of the dictyostelium. Dictyostelium are model organisms for cellular slime molds, so they're often studied quite extensively. Now, they can reproduce sexually and asexually. This here shows the asexual reproduction, and of course, asexual reproduction often is done with the haploid cells. Okay, so here we have, let's start with the migrating aggregate. So here we have a bunch of amoeboid cells which have that they've aggregated and they've formed into something that can move. So this migrating aggregate occurs when the conditions are very poor and it needs to find a good food source. So it moves, it finds a good food source, and then it hangs out there. So once it finds a good food source, it's able to produce these fruiting bodies, which are eventually going to produce spores. So these are sporangium, right? So they're going to produce spores, and this is going to occur by mitosis because they're already haploid and the spores are haploid. So it has to be mitosis in order to produce the spores. Once the spores land somewhere, they're going to germinate, I guess, if you will. Basically, what's going to happen is an amoeba cell is going to emerge from the spore coat, right? So the surrounding material, which protects it from adverse conditions. And then, of course, these are the feeding stage similar to the Plasmodial slime mold. However, they are individuals, so they feed on their own and reproduction. So what can happen with these amoeba is that they can fuse together, and this is fertilization, and then they create a zygote, which is diploid. The zygote is going to, or the cells are going to undergo meiosis in order to produce haploid amoebas. And this is going to result in increased variation, as any method of sexual reproduction does, because different amoeba or genetically different amoeba can fuse together. So then you end up with haploid amoeba that are different from the original two amoeba. Now the tubulinids are quite diverse. They are amoebozoans, and they do have lobe or tube-shaped pseudopodia. They are very common unicellular protists that are found in soil and water. Most tubulinids are heterotrophic and actively seek bacteria and other protists to consume. So this is probably the one you are most familiar with. This is Amoeba proteus. And you can see that it does have these lobe-shaped or tube-shaped pseudopodia. And you can see the nucleus here. So this is one that's commonly studied or shown as a model example for really just the general term amoeba. But you can see now that there are different types of amoeba or amoeboid type cells. Entamoebas, and yet another one, are actually parasites of vertebrates. And some of them are parasites of invertebrates. Entamoeba histolytica causes amoebic dysentery in humans, and it's actually apparently the third leading cause of death due to specifically eukaryotic parasites. Now moving on to the major group of Opisthokonts. Okay, so this is a major subgroup of the Unikonta. And again, the Opisthokonts include the animals and the fungi, and then, of course, the groups of protists that are most closely related to each one of them. So here we can see the nuclearid. The nuclearid is actually the sister, it shows a sister group to fungi. So this is the This is basically the direct ancestor of fungi, or at least they shared a direct ancestor, so they have a common ancestor. And then we have the choanoflagellates, which are the sister group to animals, and those two shared a common ancestor directly. So the choanoflagellates are most closely related to animals. There really isn't much information in your textbook about these groups. And they'll probably be mentioned again when we get to animals and fungi, those chapters respectively. This is a summary of the Unikonta. So remember, Unikonta divide into the two groups, Amoebozoans and the Opisthokonts. The Opisthokonts include those ancestors of animals and fungi, and the Amoebozoans further divide into slime molds, tubulinids, and the Entamoebas. And remember, the slime molds, there are Plasmodial slime molds and cellular slime molds.